Mobile genetic elements and viruses. We have seen that homologous recombination can promote the exchange of DNA sequences between chromosomes, but such exchanges are generally conservative. The order of the genes on the recombined chromosomes remains the same, because homologous recombination occurs only between chromosomes that are very similar in sequence. But genomes are also subject to more dramatic forms of genetic variation. Changes that alter the order of genes on a chromosome or even add new information. It is this more radical form of genetic resculpturing we turn to next. The agents underlying these drastic genetic changes are mobile genetic elements, sometimes known informally as jumping genes. Found in virtually all cells, these elements are short, specialized segments of DNA that can move from one position in the cell's genome to another. Although they can insert themselves into virtually any sequence within the genome, most mobile genetic elements lack the ability to leave the cell in which they reside. Their movement is therefore restricted to a single cell and its descendants. This is not the case for viruses, essentially just strings of genes wrapped in a protective coat. Viruses are the ultimate mobile DNA in that they can escape from one cell and infect another. We end this section with a brief discussion of viruses, which, although they are occasionally beneficial to cells, are also responsible for some of our most devastating diseases. Mobile genetic elements encode the components they need for movement. Unlike homologous recombination, the movement of mobile genetic elements does not require DNA sequence similarity. Instead, each element typically encodes a specialized recombination enzyme that mediates its movement. Figure 6.32. These enzymes recognize and act on unique DNA sequences that are present on each mobile genetic element. Many mobile genetic elements also carry other genes. For example, mobile genetic elements that carry antibiotic resistance genes have contributed greatly to the widespread dissemination of antibiotic resistance in bacterial populations. Mobile genetic elements are also called transposons and are typically classified according to the mechanism by which they move or transpose. In bacteria, the most common mobile genetic elements are the DNA-only transposons. The name is derived from the fact that during their movement, the element remains as DNA, rather than being converted to RNA as for other elements that we discuss below. Bacteria contain many different DNA-only transposons. Some move to the target site using a simple cut-and-paste mechanism, whereby the element is simply excised from one genome and inserted into a different site. Other DNA-only transposons replicate their DNA before inserting into the new chromosomal site, leaving the original copy intact at its previous location. Figure 6.33. The human genome contains two major families of transposable sequences. Amazingly, nearly half of the human genome is made up of millions of copies of various mobile genetic elements, which form a very large part of our DNA. Some of these elements have moved from place to place within the human genome using the cut-and-paste mechanism discussed above for DNA-only transposons. See Figure 633a. However, most have moved not as DNA, but via an RNA intermediate. These are called retrotransposons and are, as far as is known, unique to eukaryotes. One abundant human retrotransposon, the L1 element, sometimes referred to as line 1, is transcribed into RNA by the host cell's RNA polymerases. A DNA copy of this RNA is then made using an enzyme called reverse transcriptase, an unusual DNA polymerase that can use RNA as a template. The reverse transcriptase is encoded by the L1 element itself. The DNA copy of the element can then reintegrate into another site in the genome. Figure 6.34. L1 elements constitute about 15% of the human genome. Although most copies are immobile due to the accumulation of deleterious mutations, a few still retain the ability to transpose. Their movement can sometimes result in human disease. For example, about 40 years ago, movement of an L1 element into the gene that encodes factor VIII, a protein essential for proper blood clotting, caused hemophilia in an individual with no family history of the disease. Another type of retrotransposon, the ALU sequence, is present in about 1 million copies in our genome. ALU elements do not encode their own reverse transcriptase and thus depend on enzymes already present in the cell to help them move. Comparisons of the sequence and locations of the L1 and ALU elements in different mammals suggests that these sequences have proliferated in primates relatively recently in evolutionary time. Figure 635. These highly abundant elements scattered throughout our genome must have had major effects on the expression of many of our genes. It is humbling to contemplate how many of our uniquely human qualities we might owe to these genetic parasites. Viruses are fully mobile genetic elements that can escape from cells. Viruses were first noticed as disease-causing agents that, by virtue of their tiny size, passed through ultra-fine filters that can hold back even the smallest bacterial cell. We now know that viruses are essentially genes enclosed by a protective protein coat. However, these genes must enter a cell and use the cell's machinery to express their genes to make proteins and to replicate their chromosomes. Virus reproduction is often lethal to the cells in which it occurs. In many cases, the infected cell breaks open, lyses, releasing the progeny viruses, which can then attack neighboring cells. Many of the symptoms of viral infections reflect the lytic effect of the virus. The cold sores formed by herpes simplex virus and the blisters caused by the chickenpox virus, for example, both reflect the localized killing of skin cells. Although the first viruses that were discovered attack mammalian cells, it is now recognized that many types of viruses exist. Some of these infect plant cells, while others use bacterial cells as their hosts. Viral genomes can be made of DNA or RNA and can be single-stranded or double-stranded. Table 6.2. The amount of DNA or RNA that can be packaged inside a protein shell is limited, and it is too small to encode the many different enzymes and other proteins that are required to replicate even the simplest virus. For this reason, viruses must hijack their host's biochemical machinery to reproduce themselves. Viral genomes typically encode the viral coat proteins as well as proteins that co-opt the host enzymes needed to replicate their genome. Figure 636. The simplest viruses consist of a protein coat made up primarily of many copies of a single polypeptide chain surrounding a small genome composed of as few as 3 genes. More complex viruses have larger genomes of up to several hundred genes, surrounded by an elaborate shell composed of many different proteins. Figure 637. Retroviruses reverse the normal flow of genetic information. Although there are many similarities between bacterial and eukaryotic viruses, one important type of virus, the retrovirus, is found only in eukaryotic cells. In many respects, retroviruses resemble the retrotransposons we discussed earlier. A key feature of the life cycle of both of these genetic elements is a step in which DNA is synthesized using RNA as a template. The retro prefix refers to the reversal of the usual flow of DNA information to RNA. See Figure 7.2, and the enzyme that carries out this step is a reverse transcriptase. The retroviral genome, which is single-stranded RNA, encodes this enzyme, and a few molecules of the enzyme are packaged along with the RNA genome in each virus particle. The life cycle of a retrovirus is shown in Figure 638. When the single-stranded RNA genome of the retrovirus enters a cell, the reverse transcriptase brought it in. brought in with it makes a complementary DNA strand to form a DNA/RNA hybrid double helix. The RNA strand is removed, and the reverse transcriptase, which can use either DNA or RNA as a template, now synthesizes a complementary strand to produce a DNA double helix. This DNA is then inserted or integrated into a randomly selected site in the host genome by a virally encoded integrase enzyme. In this state, the virus is latent. Each time the host cell divides, it passes on a copy of the integrated viral genome, which is known as a provirus, to its progeny cells. The next step in the replication of a retrovirus, which can take place long after its integration into the host genome, is the copying of the integrated viral DNA into RNA by a host cell RNA polymerase, which produces large numbers of single-stranded RNAs identical to the original infecting genome. The viral genes are then expressed by the host cell machinery to produce the protein shell, the envelope proteins, and reverse transcriptase, all of which are assembled with the RNA genome into new virus particles. The human immunodeficiency virus, HIV, which is the cause of AIDS, is a retrovirus. As with other viruses of this type, the HIV genome can persist in the latent state as a provirus embedded in the chromosomes of an infected cell. This ability of the virus to hide within host cells complicates attempts to treat the infection with antiviral drugs. But because the HIV reverse transcriptase is not used by cells for any purpose of their own, this enzyme is one of the prime targets of drug development against AIDS. Essential concepts. The ability of a cell to maintain order in a chaotic environment depends on the accurate duplication of the vast quantity of genetic information carried in its DNA. Each of the two DNA strands can act as a template for the synthesis of the other strand. A DNA double helix thus carries the same information in each of its strands. A DNA molecule is replicated by the polymerization of new complementary strands using each of the old strands of the DNA double helix as a template. Two identical DNA molecules are formed, enabling the genetic information to be copied and passed on from cell to daughter cell and from parent to offspring. As a DNA molecule replicates, its two strands are pulled apart to form one or more Y-shaped replication forks. DNA polymerase enzymes situated at the fork produce a new complementary DNA strand on each parental strand. DNA polymerase replicates a DNA template with remarkable fidelity, making less than one error in every 10 to the power of 7 bases read. This accuracy is made possible in part by a proofreading process in which the enzyme removes its own polymerization errors as it moves along the DNA. Because DNA polymerase synthesizes new DNA in only one direction, only the leading strand at the replication fork can be synthesized in a continuous fashion. On the lagging strand, DNA is synthesized in a discontinuous backstitching process, producing short fragments of DNA that are later joined by the enzyme DNA ligase to complete the DNA strand. DNA polymerase is incapable of starting a new DNA chain from scratch. Instead, DNA synthesis is primed by an RNA polymerase called primase, which makes short lengths of RNA primers that are elongated by DNA polymerase. The primers are subsequently erased and replaced with DNA. DNA replication requires the cooperation of many proteins. These form a multi-enzyme replication machine that catalyzes DNA synthesis. In eukaryotes, a special enzyme called telomerase replicates the DNA at the ends of the chromosomes. The rare copying mistakes that slip through the DNA replication machinery are dealt with by the mismatch repair proteins. The overall accuracy of DNA replication, including mismatch repair, is 1 mistake per 10 to the power of 9 nucleotides copied. DNA damage caused by unavoidable chemical reactions is repaired by a variety of enzymes that recognize damaged DNA and excise a short stretch of the DNA strand that contains it. The missing DNA is resynthesized by a repair DNA polymerase that uses the undamaged strand as a template. Non-homologous end joining allows the rapid repair of double-strand DNA breaks. The process often alters the DNA sequence at the site of the repair. Homologous recombination can faithfully repair double-strand DNA breaks using a homologous chromosome sequence as a guide. During meiosis, a related homologous recombination process causes a shuffling of genetic information that creates DNA molecules with novel sequences. Mobile genetic elements, or transposons, move from place to place in the genomes of their hosts, providing a source of genetic variation. Nearly half of the human genome consists of mobile genetic elements. Two classes of these elements have multiplied to especially high copy numbers. Viruses are little more than genes packaged in protective protein coats. They require host cells to reproduce themselves. Some viruses have RNA instead of DNA as their genomes. One group of RNA viruses, the retroviruses, must copy their RNA genomes into DNA to replicate.